So it's my pleasure to introduce our speaker today, Stephanie Jones from the Department of Neuroscience at Brown University. So Stephanie, as you will see in her talk, she has kind of an unusual background, and she brings that to all the neuroscience she does. She has a bachelor's in mathematics with a minor in computer science from Boston College. Correct me on any of these things. I'm going to do it all from memory.
So far, so good.
She went on to do her PhD work at Boston University with Nancy Capel, who many of us know, a leader in dynamical systems related to brain function. And really, Stephanie embodies that, and she brings this brand of mathematics and dynamical systems analysis. But the important thing is she ties it to the underlying circuitry in the brain. And so she's taken that through work she's done following her PhD, worked at the Martinez Center at Harvard Medical School, where she got involved in doing human MEG and EEG, these large sort of macroscopic measurements that are really amenable to cognitive measuring electrophysiological signals and during cognitive function. Then went on to her position, there's many things in between, went on to her position at Brown University where she's an associate professor. And I think you will see that she really does kind of capture this merging of or integration of different levels of these macroscopic signals that we measure non-invasively, often non-invasively in the human brain, to the more microscopic circuit level details that are kind of a lot of us think about. So I think you're an exemplar of that. So I hopefully we'll hear some of that today. So
Excellent.
Thank you, Stephanie, for joining us.
Thanks, Garrett. Thank you for that wonderful introduction and for inviting me to speak. I'm really excited to be here with you all and to have the opportunity to discuss my research program with this great community. So I'd like to begin by giving you an overview of my approach to studying human brain rhythms. We use MEG and EEG, which is Magneto and Electro-encephalography, to measure macro-scale signatures of human information processing. And in particular, we study somatosensory perception. Now, these techniques are really powerful in that they give us these direct neural correlates of human behavior with millisecond resolution, which is really nice. But a downside is that we're recording this macroscopic scale activity that's generated by large ensembles of neurons. And it can be really difficult to infer what's going on at the cellular and circuit level. And it's this circuit level understanding that we need if we want to connect these signals to function in a meaningful way, and if we want to target any kind of treatment when they disrupt. Now, electrophysiology, intracranial recordings, and modulation techniques like optogenetics, these are ideal for dissecting circuit level dynamics, but there's obvious difficulty in applying those in the human. And so what we have done to bridge this gap is we've been developing these biophysically principled models of neocortical circuits. And these models are designed to capture the physics underlying these macro scale signals, but with enough detail to be interpreted at this cellular and circuit level. And in keeping this model, the output of the model in very close agreement with our data, we've come up with some new hypotheses as to the origin of non-invasively measured brain rhythms and their meaning for information processing. And today I'm going to tell you about a particular rhythm known as a beta rhythm that's defined by this 15 to 29 hertz frequency band. So why am I concentrating on beta rhythms? Well, beta rhythms are one of the most prominent signatures of activity that you measure with MEG and EEG from all over the brain. And what I'm showing you here is perhaps the most well-known example of beta activity, which is beta activity from the motor cortex. So this is an earlier study by Brown and Marsden, where they're recording from an EEG sensor above primary motor cortex, and they filtered this signal in the beta frequency band. And on the left, I show you data from a healthy control subject that's holding their wrist steady or moving their wrist up and down. And you can see that during this steady hold position, you get this large amplitude beta oscillation above motor cortex. When the subject moves their wrists up and down, the beta rhythm dissipates, or what we say desynchronizes with movement. Now, in the middle, I show you a Parkinson's disease patient who has bradykinesia. And that's trouble with movement such that it's slower. And when they try to move their wrist, it's slower and lower in amplitude. And correspondingly with this difficulty in movement, there's a lack of beta desynchronization. And so in this sense, we can think about beta as signaling the inhibition of movement. And when Parkinson's disease patients are given medication that alleviates the motor symptom, it also reestablishes this beta desynchronization. So this is an example from motor cortex, but beta rhythms, as I said, are measured all over the brain, and we've been studying them in sensory cortex. And we've been studying their mechanisms in meaning in somatosensory processing. And so there's two main questions that I'm going to address in this talk. The first is, do beta rhythms in human sensory cortex influence processing? And if so, why? And as you can imagine, the answer to the first question is yes. And I'm going to show you several lines of evidence that suggests that pre-stimulus beta rhythms in this spontaneous period before a stimulus can in, predict inhibited perception. And then the more interesting question is why? Why is it when that the circuits are producing beta, we have a lack of sensory perception, we have a lack of motor movement? And to address this question, we're going to turn to our model. And we're going to study with our model, well, what are the circuit mechanisms that create this beta, and how do those mechanisms translate to inhibited function? But before we can study the mechanisms of beta, we have to take a step back and understand better what it is that we're trying to model. And to do that, there are some key additional questions that we have to ask about our data. And the first question is, what does beta look like in unaveraged data? So the majority of studies, including our own, that correlate beta rhythms with function, we average. We average across trials. We average across the beta frequency band. We average across time. And then we say, oh, this average power correlates with this or that function. But the brain isn't averaging across trials. And with our model, we don't want to reproduce the average signal. We want to understand what is the signature of this beta activity at any moment in time. And then once we know what it looks like on average data, we can then ask, well, what features contribute to the functionally relevant differences in average power? Once we know what those features are, we can study with our model the circuit mechanisms that create those specific features. And then we can ask, how do these mechanisms translate to inhibited function? And so in stepping through answering each one of these questions with our integrated approach, the punchline that I'm going to arrive at is that we've come up with a new theory on the circuit level dynamics that create beta. And this theory further predicts that beta decreases the salience of sensory input via the recruitment of layer-specific inhibition. And this inhibition decreases the relay of information throughout the brain. Okay, so I'm going to take you through many steps to get to this final punchline. So the first question, do beta rhythms in human sensory cortex influence processing? Well, I'm going to be concentrating on our MEG. And as I've said, we study somatosensory information processing. And our main interest has been in signals from the primary somatosensory cortex. So we take this whole head imaging data, and we apply what's called a standard inverse solution method, and we isolate the contribution to the signal from S1. And so what I'm showing you here is a source in the hand area of S1, and just looking at the spontaneous activity in that area when the subject is just sitting in the MEG scanner with their eyes open. And what you see is this large amplitude, low frequency oscillation. If we put a frequency filter on this signal, this is just a standard wavelet analysis. Here's frequency, here's time. You see that there's a strong beta component in this rhythm, this 15 to 29 hertz band, but emerges as part of this complex rhythm that has both alpha and beta activity. Now this is the signature of activity that we see from every adult subject that we put in the scanner. Everybody shows these really large alpha and beta oscillations. Everywhere in the brain, what you really get with these techniques is these low frequency, large amplitude oscillations. So whether you think that rhythms matter or not, it's highly debated out there whether rhythms are causally important. This is what we have in the human. So this is the signal that we're working with, and so in our view, it's important to understand what it means, even if it's not the direct causal implication for function. So the question we asked were, at any moment in time, the expression changes, and we ask, does the spontaneous expression impact the subject's ability to perceive a tactile stimulus? And so the first task that we ran was a tactile detection task. And let me just briefly describe the task. Subjects are seated with their eyes open. They're staring at a fixation cross. And the trial begins and ends with an auditory mask. It's just a tone, doot, and then it goes off. Randomly within that tone, they're given a brief tap to the middle fingertip. The strength of that tap is maintained so that they're always feeling it half of the time. They start to feel it, we make it weaker or stronger. And so we have a perceptual threshold level stimulus. And then they respond whether they felt it or not with a button press of their opposite hand. And we ask the question, do the beta rhythms in this pre-stimulus period, before that tap comes in, predict whether the subject will feel it or not? And the answer is yes. The beta component in particular was a beautiful predictor of tactile detection, such that if you had more beta power, you were less likely to feel that tap to the finger. So what I'm showing you here is we take our trials, we take the 1 second pre-stimulus period, and we average. We average across time. We average across the frequency band. And then we sort that average power from low to high. And we say, well, if it was low power, what was the probability of detection? And here's detection rate, which is a % change in hit rate from the mean hit rate. And you can see that the lower the average beta power, the more likely you are to feel that tap. And so in this sense, beta seems to be inhibiting the perception of the stimulus. Now, this was just a detection test, but presumably, on trials where they felt the stimulus, they were paying more attention to their finger on that trial. So the next study that we ran was a cued attention task. We said, okay, now pay attention to a stimulus in either your foot or your hand, and let's look at what the rhythms do after that attentional cue. So here is the task. The subjects are again seated, eyes open. A visual cue comes on that says, pay attention to stimulation in your hand or your foot. Randomly, the stimulation was delivered to either the big toe or that middle finger. And you had to respond whether you felt it in the attended location. So you have to both actively attend to where you're cued and block anything that might be distracting in the opposite location. And we looked at this post-cue, pre-stimulus period and said, does attention modulate the rhythms? And we found, again, that it has a strong impact on beta power. So here I'm showing you the signal from the hand area of S1. When the subjects are cued to attend to their hand, the power of these rhythms decreases, suggesting these rhythms get out of the way for optimal information processing. Now, not only did we look at power in this primary sensory area, we also looked at coordination with attentional control, executive control networks in frontal cortex to see, well, was there any coordination of this decrease in beta power with attention? And it turns out, in fact, there was. So here I'm showing you coherence, again, between the hand area of S1 and regions in the frontal cortex. And we're looking at here, coherence is phase locking value, the phase locking of these beta rhythms after that attentional cue. And we can see that after the cue, in the beta band, the coherence decreases with attention. Again, suggesting there's some mechanism to get these out of the way. And this was specific to the right inferior frontal cortex, which fits with many prior studies about the role of this part of frontal cortex in attentional control. So, in answer to the first question, do beta rhythms in human sensory cortex influence perception? Absolutely. They correlate with function. Similar in motor cortex, their presence suggests inhibited function, inhibited perception. So now the more interesting question, why? What is it about beta that translates to, I didn't feel it, I couldn't move? And again, we have to first step back and say, if I'm going to use so we're going to turn to our model, and we're going to simulate this, but what are we modeling? We have to have an accurate representation of our data that if we reproduce it in our model, we say, yes, this is a good model of beta because it matches our data. So let's look at our data. What we find when we look at unaveraged data is that these spontaneous beta rhythms are very transient. They last about 150 milliseconds or less, on average about 120 milliseconds. What I'm showing you here is single trial data. So this is 10 examples of 1 second of pre-stimulus data. And here's the raw waveform, and here's the frequency spectrum. And I've highlighted with these red boxes these beta events. And they're very transient. Again, about 120, less than 150 milliseconds. Now, in the frequency domain, these signals are always positive. This is 0 and above. So that when you average these accumulate without cancellation. It's not like the time domain where you have positive, negative signals and they cancel. They don't cancel. And so in the average, you get this beautiful band of activity in the beta band. And your mind automatically conjures up, oh, I had this nice continuous oscillation. But in fact, that's not what you have here. And so that's not what you want to reproduce with your model. We can take the average across time and get more of a parallel spectral density. And it's this sort of thing, or something like this, that 99% of the studies are looking at when they're correlating beta with function. We've looked at many other data sets, and this transient nature of beta seems to be a very robust phenomena. So let me show you some of our other data sets. Here on the upper left, I'm showing you the MEG data from frontal cortex in that same data set where I showed coherence with sensory cortex. And you can see in this data, and on average, beta emerges as these brief events. Over here in the upper right, I'm showing you some data from an ECOG sensor over sensory motor cortex. So this data was provided by my collaborator, Dr. Wael Assad, who's a neurosurgeon at Rhode Island Hospital. And he implants deep brain stimulating electrodes in thalamus. But while he's doing the surgery, he also puts these ECOG strips over frontal and sensory motor cortex. And we just looked at the spontaneous activity and found remarkably similar transient beta events in this signal. Down here, I'm showing you data from local field potential recordings. This one in anesthetized mice from my collaborator at Brown, Christopher Moore. This is in the somatosensory cortex of mice. And over here in awake monkeys, this was data provided by Charlie Schroeder at the Nathan Klein Institute. So the punchline here is beta is transient. There's other recent publications by Anne Grable's group that shows a similar thing in the striatum and motor cortex of awake behaving monkeys. Now, one thing you might notice is I've put a red box now around the waveform. And if you look closely, it seems like there may be some stereotypical waveform that's underlying these big beta events. And so we looked more closely at that in our MEG data. And what we found was that these beta events do indeed have a stereotypical waveform. And so what I'm showing you here is just a single trial. We look for these high-power beta events in the frequency spectrum, and then we go back to our raw data, and we say, what does this raw signal look like during this beta events? And here I'm showing you 50 single high-power beta events in the average. And you can see that there's something very stereotypical happening here. We've quantified different features of this waveform. I'm not going to go into the details because I think it's pretty obvious that the dominant thing here is this large amplitude negative deflecting peak, which we call PK3. Now, we've looked at this in different subjects, so this is a single subject. This is the data from 10 different subjects. And while the amplitude of the oscillation varies, the overall waveform is consistent. And the important thing here is that the reason that this turns out to be beta in the frequency domain is because this main peak lasted 50 milliseconds. So we had some big event, it lasted 50 milliseconds, we put a frequency filter on it, and we got a beta event. This is not a sinusoidal oscillation that has a 50 millisecond period. And that's very important when you're thinking about the mechanism that can create something like this. Okay. So what does beta look like? Well, it's event-like, and it has a stereotypical waveform. So now we can ask, what features underline these differences, these functionally relevant differences in average power? Now, typically when you think about differences in power, again, at least for me, your brain goes to a sinusoid. You have an oscillation. And you think, oh, if I have higher power, I have a larger amplitude oscillation. That means more of my network participated, or there was more synchrony or something going on. And that's kind of intuitive to what we think. But given that these things are transient, there are several possible features that could underlie differences in average power. One thing that could happen, so here I'm showing you again frequency and time, and this is a representation of the average power across that frequency and time window. It could be, here's one event, that would give low power. It could be that I have two events in that time window, right? And I average this and I get low power, but I average this and I get higher power. It could be that the amplitude of these events was bigger. In this case, again, I'd get low average power. In this case, I'd get high average power. It could be that these events get longer in duration, or it could be that they get longer in frequency span. Any one of these could underlie the functionally relevant differences that we see. And so we ask the question, which is it? Let's look in our pre-stimulus period, and let's see what correlates best with average power: event number, amplitude, length, or frequency span. And what we found, first thing we have to do, let me briefly describe how we define an event. So we have to somehow say this is an event, this is an event, right? Because we see these high power blobs that come about, but there's also some low power things in here too. And so we want to be able to say this was an event, this wasn't. How do we calculate it? So we can threshold, and we can look at the power. Here's 3 different thresholds calculated as a factor of the median power. And we see how well does the power above this threshold account for the power in the entire window. And it turns out that the thresholding doesn't matter that much. No matter which one we pick, we get a very high correlation with the average power. That's because these events are dominating the signal. As I lower the threshold, I'm just getting more and more blue. I'm adding more and more 0 to my average. And so it's not really changing the correlation with power. And so for the rest of what I'm going to show you, we're just looking at one threshold, and it happens to be this highest threshold here. Okay, so now let's threshold our spectrogram, look at our events, and see, is it the number, amplitude, length, or frequency span? And it turns out it's the number of events that shows the strongest correlation with pre-stimulus average power. Here is, this is the mean power on any trial sorted from low to high and the corresponding number of events in that pre-stimulus time window. It's, again, 1 second pre-stimulus. Here's the power and the amplitude of the events, the length of the events and the frequency span. And this is the Pearson's correlation coefficient. And you can see very clearly it's the number of events that has the most correlative predictive behavior of power. Now, this is in our S1 detection task. We've also looked at this in our attention task. So now, rather than looking at the pre-stimulus period in our detection task, we're looking at that post-cue pre-stimulus period in the attention task. We see a very similar phenomena. We've also looked at this in the mouse data. So again, this is through my collaborator, Chris Moore, who's doing whisker detection in mice and recording local field potential signals in the primary somatosensory cortex. And I've already shown you that in the local field potential, these things are also event-like. And in this signal as well, it's the number of events that's predictive of average power. Okay, so that's just average power. But the more important question is it predictive of perception? So now, rather than sorting by power, let's sort by perception. Did you feel it or didn't you feel it, a hit or a miss? Let's sort by attention. Were you attending or not attending? And let's see what happens in the pre-stimulus period. Is there a difference in the number of events or some of these other beta features? And it turns out that only the number of beta events is higher in non-detected and non-attended trials. Okay, so remember, in non-detected trials, you have higher power. The question in non-attended trials, you have higher power. The question we're asking is, what's contributing to that? Is it that you had more events, they got bigger, longer, or increase in frequency span? And what this is showing is that you had more events. So here I'm sorting data over hit and miss trials and looking at the number of events in each of these things, and you can see the only significant differences here. We looked at the same thing in our attention data. Here we're sorting over attend in and attend out, and you get more of these beta events in the attend out condition, and it's similar in the mouse data. So this is a different way to think about how these rhythms change with perception and power. What seems to be happening is when you say pay attention to your finger, what the brain is doing, it's decreasing the probability that this thing happens. And that decrease in probability that this thing happens corresponds to an increase in function. Now, that's just looking at the number, but then you could ask the question, well, what about the timing? Does the timing of when this event happened right before the stimulus came in, does it matter? And in fact, the timing matters as well. So here I'm showing you the timing of the most recent beta event. So 0 here is the time of the stimulus. Okay? And we look in the pre-stimulus period and say, when was the last event? This would be a recent event. This would be a less recent event. It turns out that non-detected trials have more recent beta events. The closer it comes before the stimulus comes, the less likely you are to feel it. And so there's a timing effect here as well. And again, remarkable consistency across species in recording modalities. Okay, so what features underlie the functionally relevant differences in average power? Well, it's the number of beta events is greater during non-attended, non-detected trials, and non-detected trials have more recent beta events. So now we can say we've quantified what it is we want to understand with our model. Now we can turn to our model and say, what are the circuit mechanisms that can create beta events that look like this and may underlie this difference in perception? So in building a model to study these signals, we have to first consider very carefully, what is it that we're trying to model? We're trying to model these macro-scale MEG and EEG current sources. So when we take our data and we apply what I've told you is called an inverse solution technique to isolate the location and temporal profile of the signal, what we're doing is we're estimating the primary electrical currents. These primary currents come from the postsynaptic intracellular subthreshold current flow in the pyramidal neuron dendrites. Essentially, these long pyramidal neurons, they're long, they're oriented like telephone poles lined up. The net electrical current within them is large enough and synchronous enough to produce an electrical current in a magnetic field signal that can be recorded outside of the head. So in our model, this is what we want to be able to simulate. And so to build a model that could simulate this intracellular electrical current flow, we've created this laminated model of a cortical circuit. It contains synaptically coupled multi-compartment pyramidal neurons in the supragranular and infragranular layers, and inhibitory interneuron shown here in red in the supragranular and intergranular layers. We also have inputs from other parts of the brain. So this is representative of primary somatosensory cortex here. We have input that comes in from the lemniscal thalamus. This is the thalamic nuclei that when you have sensory information, it goes through the thalamus and up into the cortex and right into the granular layers, and then effectively will contact the pyramidal neuron, the proximal dendrites of these pyramidal neurons. We include another input that goes right up to the supragranular layers. Now this input would be the input that comes in either from a higher order cortical area or from a non-lemniscal thalamic nuclei that projects right up to the supragranular layers. And for these spontaneous rhythms, we're thinking that both of these inputs probably come from thalamus, although we don't know that. In the model, all we do is we generate trains of action potentials. And these action potentials drive postsynaptic receptors in our cortical network in different projection patterns. So we simulate the electrical activity of these neurons with Hodgkin-Huxley current balance equations. We have active dendrites, active somas, and then we calculate the net intracellular electrical current flow. It's a cable equation, right? And that net intracellular current flow is what we compare to our MEG data. And with this construction, the units of measure that come out of the model are nanoampere meter, current times distance. That's exactly what we get out of our MEG data. And so we have one-to-one comparison between our model and our MEG, and this allowed us to make new predictions about where these rhythms come from. Now, an important thing to emphasize is that if we tried to reproduce this rhythm with more simplified models, this mechanism would have been missed. There are a lot of ways beta rhythms could come about in neural circuits. But it wasn't until we included the right biophysical properties of the neurons and what contributes to the physics of the MEG and EEG signal that all these new predictions came about where these rhythms can come from. And what I've been showing you here is a reduced representation of the model. In fact, the model represents a cortical column. There's hundreds of pyramidal neurons in the supragranular and infragranular layers. The unique thing about our model is that it's the only model that simulates human current source signals from intracellular current flow. We simulate it in a software package called Neuron. It's compatible with Python. And we just recently got an NIH Brain Award to turn this model into a tool that other researchers doing MEG or EEG or even ECOG can start to use to make predictions about the circuit level origin of their imaging data. So we're very excited about that. Okay, this is a very complicated model. Thousands of differential equations. Hundreds of cells, different layers. How do we constrain the madness? There is a method to the madness. And our method was as follows. the first thing that we did is we spent a lot of care to fix the cellular physiology. We built the morphology and the physiology, the active currents in these neurons, so that they matched what's been recorded in vivo, primarily from literature from mice and primates and somatosensory cortex, of which there's a lot of literature out there. So we built this in, and then we fixed all those parameters. Then we looked at the local connectivity. Who should talk to which layer, and what should the synaptic time constants be? Again, large body of literature, did the best we could, and then we fixed it. The only parameters that we're tuning to simulate these rhythms is the exogenous drive to the local network. We tune the timing and these strengths of these driving spikes that come in what we're calling a proximal projection pattern because it hits the proximal dendrites, and a distal projection pattern because it hits the distal dendrites. So how do we tune these knobs to reproduce these S1 beta events? Well, remember what we're trying to model. An accurate model should reproduce this waveform. So that's what we're going for here. And again, it's not this. This lasts 120 milliseconds. So does this oscillation here. Both of these, if you put a frequency filter on them, will give a brief beta event. Both of them. But the mechanism that can create this versus this, very, very different. And so that's one thing that I really want to emphasize is that the human imaging literature, EEG, MEG, ECOG, we have to take a little step back and stop averaging and making conclusions about what these things mean and take a close look at our data. Okay, so through a long history of simulations, we have lots of publications on this. We arrived at the following prediction, and I'm just going to give you the punchline, which is this. To reproduce this beta event in the model, so this is now all model, what we did is we took our circuit and we drove it with two inputs, one in the proximal projection pattern and one in the distal projection pattern. And when the proximal input was broad and weak, and the distal input was strong and lasted 50 milliseconds, the net current, which is this intracellular current, created our beta event waveform. And so I can unpack that for you a little bit over here, where on the bottom I'm showing you a histogram of the spikes that drive the network in this proximal projection pattern. On the top I'm showing you the histogram of spikes that drive in this distal projection pattern. So if I only had these proximal inputs, what this does is it comes in, oops, sorry, what happened there? There it is. And it drives current flow up the dendrites, right? So the proximal input comes in, it's hitting the proximal dendrites, it pushes current flow up the dendrites. And so if I only had this proximal input, I would just get one big broad bump here. But at the same time, I'm giving it this strong distal input. This distal input is driving current flow down the dendrites. And so it pushes back, drives current flow down the dendrites. If it lasts a beta period, then this lasts a beta period, and I get a beta oscillation in my waveform. And so this is our beta mechanism. Two inputs, this one's strong and lasts 50 milliseconds. Now, we know that in S1, these beta oscillations don't occur in isolation, right? They occur as part of this alpha-beta complex. And another thing that we've shown, and again, I'm not going to go into a lot of details, this has been published, is that if we do these two inputs to be rhythmic, so that they come in every 100 milliseconds, so I do this every 100 milliseconds, or at 10 hertz. And there's a little stochastic nature in each cycle of the input. This one might be a little stronger on one cycle, the timing may be a little bit jittered, so there's some jitter in the timing of these inputs. Then we can reproduce the more global features of this alpha-beta complex. And this idea that in these spontaneous states, these inputs may be coming in at 10 hertz fits with the literature that suggests that in non-attentive states or non-behaving states, the thalamus is bursting at 10 hertz. There's a lot of evidence for this. And there's even some evidence from whole cell recordings that these bursts may last 50 milliseconds, which fits perfectly with our generation of beta in our model. Okay, so again, here's our prediction. Beta events come from the convergence of these inputs. This one's strong and lasts 50 milliseconds. With this, we have a very nice, and we've done some statistics to show that these are equivalent. We can reproduce our data. But the real test of whether this is the mechanism requires recordings inside the brain. And so through collaboration, we've been able to now test this mechanism invasively. So how do we, in animal models, in both mice and monkeys, so how do we connect a big macro scale current dipole signal from MEG with something that we can record with a local field potential? Well, if you remember what these MEG signals are representing, they're these big current dipoles, right? They come from the intracellular current flow in these long, spatially extended pyramidal neuron dendrites. This will produce a return current that can be picked up as a local field potential signal by a microelectrode and will go in the opposite direction. So the prediction, if we have the same beta generating mechanism, would be that, sorry, this is a little grainy. If our current dipole looks like this, then the local field potential signal from the granular layers should look like this. It should have the same shape, but it should go in the opposite direction. And so the first thing that we did, again in collaboration with our colleagues, first collaboration is with Chris Moore at Brown. He put laminar recordings I'm sorry, this is anesthetized mice. We've looked at it in awake mice as well, but this data is from anesthetized mice. Taking the local field potential signal out of the granular layer, going into the data and pulling out the high power beta events, just like we did in our MEG data. Same exact MATLAB code. And we pull out the beta events and we look at their waveform. And lo and behold, these high power beta events have a very consistent waveform. We did the same thing with data that was provided by laminar recordings collected in Charlie Schroeder's lab at the Nathan Klein Institute. These are awake behaving monkeys. Same thing, pull out the high power beta events. And remarkably, the waveform is consistent, which suggests the beta generating mechanism may be the same across species and across these recording modalities. The next test that we did is we looked at the synaptic current flow. Now remember, the model has a very specific prediction that these beta events are really driven by this strong input to the distal dendrites that lasts 50 milliseconds. And it's convergent with this broader proximal input. So the way we tested that is, again, we take our laminar recordings, we take the second spatial derivative to get a current source density measure. What the current source density tells you is current flow in and out of the cells. An excitatory synaptic current represents current flow in. We call that a sink. It's red. Current flow in, sink, red. Current flow out is a source. We call that blue. So what the model would predict is that during one of these beta events, there should be a strong sink in the supragranular layers that lasts 50 milliseconds. Okay? So this is the predicted CSD from the model, dominated by this strong sink that lasts 50 milliseconds. So we go into our animal data. Again, we take the high power beta events, and we look at what is the current source density profile around those beta events. And remarkably, again, there's consistency with our model predictions. That during these beta events, which I'm showing you here, we get this strong sink that lasts a beta period. And so this is initial confirmation that these predictions that we're getting out of the model are holding up with our invasive recordings, which is, as you can imagine, super exciting for someone that builds neural models. Okay, so now we've answered the question, what are the circuit mechanisms underlying these differences? Beta events emerge from a broad proximal excitatory synaptic drive that's simultaneous with a strong distal drive that lasts 50 milliseconds. So now to the ultimate question. How do these mechanisms translate to inhibited perception? So the way that we're investigating that is we're looking at how does beta filter that sensory input. And so what I'm showing you here is the sensory evoked response. So when we tap the finger, we get an ERP or an ERF. This is MEG data. Right? And this is the early evoked response from that tap to the finger. And what I've done is I've taken the data and I've sorted over trials where I had high pre-stimulus beta power, again in the 1 second period, or low pre-stimulus beta power. And what you can see is beta has an impact on this M70 peak. Now, one of the first studies that we did back in 2007 showed that this peak was predictive of perception. You tap the finger. If that peak is bigger, the subject says, I felt it. If it's smaller, they say, I didn't feel it. And now we're showing that high pre-stimulus beta power, which also corresponds with I didn't feel it, has a direct impact on this peak. And so the question is, why? What's going on in this circuit that's creating that effect. And why, when that happens, does the subject say, I didn't feel the stimulus, even though it clearly somehow got to the brain? So the first pass in thinking about this in the model, and I should say that all of this modeling stuff that I'm going to present now is all preliminary. We're just investigating this and trying to get a handle on what's going on in the circuit. But the first thing that we did is we said, well, during high pre-stimulus beta power, we had more events, right? It wasn't that the amplitude of these events changed. So how do we think about more events in our model? Well, events occur closer to the time of stimulus processing. So the first thing we did was we said, well, let's assume that if I'm in high pre-stimulus power, there's more likely to be an event during the stimulus. And so what we did is we simulated the evoked response either during a beta event or not during a beta event. And again, we found remarkable consistency between our model and our MEG data. And so here, I'm simulating high pre-stimulus power as simulating an event during a beta oscillation. So I do these two things at the same time. Or simulated an event not during a beta oscillation. So how do I simulate an evoked response, first of all? Well, it's a sequence of input through these same projection pathways. Right? So you tap the finger, input comes in through the lemniscal thalamus, and it drives the cortex through these proximal dendrites. That pushes current flow up the dendrites. Then about between 40 and 80 milliseconds later, input comes back from somewhere else into this local cortical circuit, pushes current flow down the dendrites, and we get this prominent N70 peak. Now the network starts spiking. These spikes drive current flow back up the dendrites, and you get this later peak, and then everything starts to die down, and to get it to start to go back up again, I have to give it another thalamic input. And there isn't perfect agreement here, but what we're really interested in is this early representation of what's going on. So why is it that if I give this sequence of sensory evoked input during a beta event, I get a lower peak that predicts perception, where without that, I get this higher peak? Well, again, we're just starting to dissect this, but what seems to be happening is that during high pre-stimulus beta, remember, beta comes from this strong input to the supragranular layers. That seems to be setting up the inhibitory network so that when I tap the finger, those inhibitory neurons go like crazy. And when they go like crazy, you get a lot of GABA driven. So this initial input comes in, things start spiking, you get a lot of GABA. When this 70 millisecond input wants to come back, I'm calling it here feedback, goes into the supragranular layers, it can't get through. And then nothing happens. And so overall, we get a low level of spiking in these pyramidal neurons that are contributing to our MEG signal. Yeah?
Clarification, so that's stimulus and not inhibition?
Yes. That's hit the network, drive inhibition, drive spikes. Mm-hmm. Okay, now if we do this without a beta event, then we see the initial input comes in, we get some spikes, but we get less inhibition. And because we have less inhibition, when this feedback comes in, we can get a lot more firing in our pyramidal neuron population. Now, the pyramidal neuron populations aren't only the populations that contribute to the MEG, but they're also the output. They go to higher order cortex, they go down to thalamus, they go down to motor centers. And so the prediction from the model is that this high pre-stimulus beta causes greater sensory evoked inhibition that decreases overall pyramidal neuron spiking and hence decreases the relay of information out of S1. And if the signal doesn't get out of S1, the subject says, I didn't feel it. And these are early. This is 70 milliseconds after I tap the finger. Okay, so let me just recap what I'm saying here. Beta is created by strong distal drive. This distal drive recruits inhibitory neurons, gets their state set so that they're ready to go. When that feedback comes in, it decreases the initial response in this M70 feedback response, and you get overall less pyramidal neuron firing. If you get less pyramidal neuron firing, you get a decrease in relay of information out of S1 and a decreased probability of perception. So we've answered our final question. How do these beta mechanisms translate to inhibited perception? Beta decreases the salience of sensory input via layer-specific recruitment of inhibition that decreases the downstream relay of information. So let me just recap our conclusions here. The first thing that I showed you was that pre-stimulus beta rhythms in somatosensory cortex predict inhibited perception. Beta events aren't these sinusoidal continuous oscillations. They're transient. They last about 150 milliseconds, and they have a stereotypical waveform. It's the number of beta events that's greater during non-detected, non-attended trial. It seems like the brain is setting up a probability that this thing happens, and that's what impacts perception. With our model, we came up with a new theory that beta comes from the convergence of these inputs, where we have one that's weak and proximal and strong distal that lasts 50 milliseconds. And this is decreasing the salience of sensory input via the layer-specific recruitment of inhibition that decreases the relay of information. Now, there's several next steps here. A lot of this is theory, predictions from the model that hasn't been tested. We have to test these theories on beta's layer-specific recruitment of inhibition with invasive recordings. And in particular, it may be that these supragranular drives are contacting these distal projection inhibitory neurons, these somatostatin positive ones that go right to the distal dendrites. We haven't answered the question, why beta, right? If we want to say beta matters for function, we want to say, oh, there's something about a 50 millisecond oscillation that matters for function. We haven't said that. All we've said is there's a 50 millisecond event and this is what it does, but we haven't said, oh, it has to be 50 milliseconds, because if it wasn't 50 milliseconds, it wouldn't work. We don't know that that's the case yet. We haven't identified the origin of this drive. We think it's the thalamus. It kind of makes sense from the literature, but we don't know that. And we want to devise improved methods to manipulate these circuits to create these beta events. And one direction my lab is going is trying to understand non-invasive brain stimulation and how we might create these events to impact perception or function. But now that we know that we're not trying to drive an oscillation, we have to think, well, how do we drive this event, and how do we drive the probability of this event? And I'm sure there's many other next steps that hopefully you guys can give me some insight on. And with that, I'd like to end and thank my collaborators. My research goals clearly require collaboration, and I feel really fortunate that I have developed a network of interactions that allow me to do this systems-level integrative neuroscience. I just want to point out a few people from my group. Rob Law is working on the modeling, Rob Law and Shane Lee, on the modeling components that I showed you, and some of the MEG data analysis. Chris Moore and Haeyang Shin have been providing the mice data. Charlie Schroeder and his postdoc Saskia Hagens provided the monkey data. I'm now working with Wael Assad and a graduate student on some human intracranial data. And my collaborator for the MEG side of things at MGH is Matti Hamalainen. So with that, I'll take questions. Somehow, some way, I got through it in time. I must have talked very fast. So, thank you.
We have time for some questions.
I know you had to move over some of the details of the modeling, but you said something interesting that I wanted to just check on, and then it leads into a second question, which is you said that the biophysical details of the model were important.
Yes.
And I wasn't quite clear from the results that you mentioned if that was a necessary component, like if you don't have Hodgkin Huxley in there, can you not? It seemed like with the possible and distal drive, you could just sort of weighted average the two and you would get something that looked like beta event.
This is a really good question. So for a beta event, I could probably have used passive dendrites. But when we think about how this impacts the sensory input, that's where those active currents matter. Because we got to get spiking. We got to get synaptic active influences induced by spiking. And even in the passive, the sub-threshold driven input, we have to have the right balance of sodium and potassium. And like if I take the potassium current out, things are too excited. And so it was important for getting the right balance of, if I give it input, will it stay sub-threshold or will it drive a spike and that sort of thing.
So then my follow-up to that is, so that's very interesting because it suggests that if you could change some of the biophysical parameters, you could change the shape of the beta event.
Yeah, that would be really interesting.
Is that, because it seems like with these complicated models, you always concern a little bit that you can explain the data, but you can predict the changes to the data.
Right.
So is there something that you can easily tweak?
So the things that we've done is taken certain currents out, right? Let's take out the sodium current, let's take out the potassium current, let's take out the M current. And the thing that seems to matter the most is the potassium currents. Because when we take them out, it's too excited. And very little input creates spiking or depolarization block. And so that's the only extent to we've looked at how do these things matter. But the period of the 50 milliseconds, that's really driven by the inputs, how long those inputs last, the cable properties of the dendrites, even that, they're not that long. You have to have current flow because that's what we're measuring. But it's really more that it effectively lasted 50 milliseconds. The time constant of excitation is hugely important. If I turn those into NMDA synapses rather than AMPA synapses, I get a very low frequency oscillation, right? Because that input is now lasting a much longer period of time.
Hello. Does your model predict tactile difficulties that Parkinson's patients have? As you said at the beginning,
Yeah.
beta, so is
That's a really good question. So we think that the same mechanism would hold for deficits in tactile perception for sure, but it could also hold for deficits in motor action because the machinery is the same, right? In motor cortex, we have a laminated cortex, we have long pyramidal neurons, we have input pathways that effectively target the proximal and distal dendrites. And so the same mechanism could be there. And the same thing could be happening in that if you have a lot of beta activity, when some signal comes from some part of the brain to say move, it can't get through because you have too much inhibition. And so you're trying to tell motor cortex to move, but the signal never gets in. And so you can never go downstream and tell your muscle to move. Same thing with somatosensory perception. So I would guess a similar mechanism would hold in Parkinson's for that reason.
So if we're going to move to motor systems, maybe you could explain to us what deep brain stimulation is actually doing.
We're actually working on that. So we have this collaboration with Wael Assad, who I mentioned, and he's doing DBS in thalamus for essential tremor patients. And so we're now working towards, and he has, before they put the DBS electrode in, they record from thalamus. And they do these interesting motor remap tasks. And so now my student Shane Lee is building a model of thalamus with a goal of trying to say, well, let's get the biophysics right here. And then can we get some novel insights about what DBS is doing and how we might titrate DBS based on the biophysics of the circuits, not just the field propagation of the electrode. Yeah.
Do you know if those short beta events are synchronized across cortical area, and especially across somatosensory and motor?
That's a really good question. We don't know. So now the first place we're looking is in frontal cortex. And so remember I said we had phase locking? Well, phase locking is, put a sinusoid on it, calculate a sinusoidal phase and look at, but now we're trying to do some more interesting things and say, well, what is it? Is it that these events happened at the same time, or what is the overlap between the two? We're just starting to look at that.
I was especially wondering about M1 since when there's motion, they seem to decrease probably in M1, and if that affects S1.
Yeah, it's really interesting. We haven't looked. It's tricky with MEG and EEG, or EEG particularly, because of the scale of these things, and it can be hard to isolate S1 versus M1, particularly in one task. It can be done, but we haven't. But it's a really interesting question.
I noticed perhaps purposeful emission from the model, which is layer 4. And there is recent evidence from mouse somatosensory cortex that layer 4 directly inhibits layer 5 and bypasses anything in the superficial layers.
Okay.
So I was wondering if you guys are thinking about including layer 4 and the effects of direct layer 4 inhibition.
We are. Right now, the only effect we have is through layer 2-3. So the input comes into 4, goes to 2-3, and then 2-3 goes down to 5. But you're right. Our initial reasoning for not including layer 4 is because it doesn't have pyramidal neurons, and so they wouldn't contribute to the MEG signal. But now that we're thinking more and more about the nuances of perception, we're thinking about adding a granular layer and more detail to the circuit. The hard thing is, it's hard to correlate that with our MEG data, right? Because we're not getting the readout from those layers. Yeah, but for
the circuit might be
right.
somewhere else. It may not be actually effective in the superficial layers.
You're absolutely right.
But
Yeah.
condition where people
Yeah, and that our trajectory has been make some predictions with the model, go back to the invasive recordings, where do things break, right? And now let's readjust our model, just like you're saying, like, oh, maybe these layer 4 things are really important and we missed something in our connection to the dipole, and then we add that layer of complexity. But that's really interesting. It might make a big difference.
We should probably ask any of our Emory friends, any questions from the Emory side? And can we hear them if they do?
Do we know there's people there?
Okay, there are definitely people there.
Okay.
They're cut off.
Hi, Emory.
Well, so I do have a question also. So presumably in the middle of all this, there's a control that says this is linked to these beta events and not just power in general. Like if you jammed it through some other battery of tests while selecting out other bands,
Yeah.
do you see any other signatures there? That,
I mean, alpha and beta are really the dominant things that we get. I mean, you can filter in gamma and say something about gamma, but it's not a gamma rhythm. It's just really the edges of these sharp amplitude oscillations. In alpha is very interesting in that with shifts in attention, alpha moves immediately. But when you correlate with perception, beta seems to be the more important thing for perception. And we look at the time course, right? We give an attentional cue, and then we look over a second. Immediately the alpha rhythms shift. But it's not until you almost get to the stimulus that the beta rhythms start to come into play. And we think that subjects learn pretty quickly, oh, I have a second before I have to think about, we gave a second before the stimulus comes in. And it's like, it's not until up to a second where they say, oh, I have to think about perception, let me engage beta. And we've seen that dichotomy of early alpha, late beta in both of our experiments. And so we don't know. The only ones that we've looked at are alpha and beta. And again, because of the scale of what we're recording.
so I think you effectively connected a lot of us who do very disparate work here. So this is a really
Excellent.
nice integrated talk. I think it covers the human sort of cognitive folks here as well as the circuit level people and everything. So thank you again for joining
All right,
us.
thank you.
Thanks, Stephanie, again.
Look forward to comments.